Did it surprise you to end up in, as part of a number of businesses where these discoveries have generated businesses that are they publicly traded, for instance? I think perhaps some of them are.
Right. So, was it a surprise that my science and technology development interests would lead to companies? That was not really surprising, at least as of when I was a graduate student, because I was already involved in consulting for companies like Biogen and Biorad when I was a graduate student. Maybe before that, I might have been a little surprised that you could even take something biological and turn it into a technology, because biotech revolution was still in the future. But being so close to the early developments in Genentech and Biogen, it seemed pretty obvious that I could do that again and again. And to some extent, it seemed like an obligation. If I'm going to develop technology, it's not adequate to just publish it in some academic ivory tower paper.
It needs to be used.
It needs to get out into the public, and that means the scientist needs to accompany it at least a little ways into the marketplace to make sure that it gets debugged and properly described.
You've brought up the idea of using CRISPR experiments to make woolly mammoth genes active again by copying them into the DNA of a modern elephant. Would that create a hybrid, or is the idea to bring back the mammoth?
Right. So we have been putting mammoth genes and other genetic changes into elephant cells to test hypotheses about what the ancient species were like. It does have the potential of making an elephant's more adapted to the modern world. It could greatly expand its range from right now. It's in really tightly confined regions where in proximity to humans, which is a bad thing for this endangered species. But while the Siberian and Canadian tundra are vast, tens of millions of kilometers squared, so it'd expand their range. That it would also allow them to help us on a potential environmental future problem that may not be of modern human cause, but could be a huge problem to deal with any other way, which is the release of carbon in the form of methane, where there's about 1,400 gigatons in the tundra that could get released, and then cold-resistant elephants could help by changing the soil temperature in various ways.
Is there anything you might anticipate learning about man from doing these kinds of experiments?
Well, all the experiments that we do on animals, no matter what their motivation, so the pigs teach us about transplantation of organs into humans, the work on dogs teaches us about aging reversal, and the work on elephants are teaching us about herpes virus, which is one of the extinction level events occurring in elephant populations today. And so in dealing with that, in dealing with making the tusks shorter, we learned something about the development of bones and teeth. The hair has all sorts of potential cosmetic applications. We learn about human biology. We develop technologies for growth of the embryos outside the body, which could be of value in preemie research and so forth. So almost every one of these animal-directed, whether it's veterinary-directed or otherwise, are a potential benefit to human medicine, and vice versa. A lot of technology we develop for gene therapy in humans, we can use in the animal projects.
I've also read that you've speculated out loud about what it might take to engineer the birth of a Neanderthal. Not necessarily that you would do it, but what's the thinking behind that?
Well, just as you can move ancient genes from animals into their modern equivalents to test the reading of their genome. So the next generation sequencing is a revolution in being able to read ancient DNA, but you need to be able to test it to see if gene A causes this ancient trait. With Neanderthals, being a close relative to humans. There are a few traits that might be interesting. You probably have to go further back to find something that's dramatically different from humans, but still in the human lineage. But these could be tested in principle in cell culture and things called organoids, where we can develop complex structures, like, for example, the larynx, which, why is it modern humans can speak so well, or brain structures that is within the near technical horizon.
What do you think is the most important thing about CRISPR?
I think I would say, ironically, the most important thing about CRISPR is that it will be replaced. It is just one of a series. It's only a small increment over its predecessor, which is TALENs, and there's lots of room for improvement in terms of precision of editing, in terms of efficiency of editing and delivery of the editing. All of those can be improved, and not necessarily in the category of CRISPR and its relatives.
So then the real question might be, what's the most important thing about gene editing? And do we even have the answer to that yet?
Well, the two most important things about gene editing, as it applies to agriculture, is that it's socially acceptable, and that it is like natural mutations. The thing that's important about gene editing for humans is that we can really do all kinds of preventative medicine, which could be called enhancement, but it's a way of anticipating as early as possible by editing. I think that could be, in the future, the biggest contribution, which is not exactly what's being the top priority right this second.